Sri Gurubhyo Namaha. Welcome to our online teaching academy Ekalaiva, and I welcome you all for the science class. This is class 10 science and we are going to see biology part. In biology already we have started with heredity and evolution and we have completed first part. Now in second part we are going to see very important thing about crosses. Before that, let us have a short recap what all we completed in the first part. So let us travel inside the cell and see what are the different parts we saw. Inside the cell you can see a rod shaped structure and that you call it as chromosome. And in the chromosome you can see again a thread like structure that you call it as DNA. Inside the DNA there is a very important thing and that is transmitting the character from one generation to the other generation. That is called gene. And another important experimental species we saw. It is nothing but Pisum sativum or pea plant. Here we saw about the 7 contrasting characters. What are the contrasting characters? Based upon the parts of the plant. They are nothing but the size, shape, color, etc. So in that they have the dominant character as well as recessive character. And the same thing, phenotype and genotype. The phenotype, the physical appearance, genotype, the gene present in that. Now we are going to deal with that only with 2 different types of crosses. And here Mendel proposed 3 different laws. We are going to see one after the other. So laws of Mendel. First law, law of dominance, then law of segregation, then law of independent assortment. 3 laws with 2 different crosses we are going to integrate. So let us see one after the other. First law of dominance, when you take, it is dominant character. Okay, only the dominant character will pass from F1 generation to F2 generation. F1, F2 as well as parent. We can take like this. From parent to the F1 generation and then from F1 to F2 also. But in F2 you can see the recessive character in less percentage. But here only the dominant character will pass from the parent to the F1 generation. Then law of segregation. Segregate, okay, something going to be segregated. Here we are going to talk about the gametes. Then law of independent assortment. Independent. What is independent? It is nothing but they are not dependent. So what is not dependent? The characters, okay. The characters show themselves, okay. They won't depend on the other characters. So here we are going to talk about the dihybrid cross, okay. And these 2 we are going to incorporate with monohybrid cross. Okay, later we are going to see what is mono, what is di. Mono a single character we are going to take. Di we are going to take 2 characters. First we will talk about the dominance. The offspring always exhibits a dominant trait. Offsprings we can talk about the F1 generation. F is nothing but filial. Okay, it's a Latin term, first generation, from the 2 alleles received from the parents. And the only dominant allele is expressed. This is very important. So from parents, the characters are getting transmitted to the next generation, that is F1 generation. Here, only the dominant characters are seen. Okay, we know the size of the plant. The size of the plant, tall. Tall will be the dominant, only the tall character will be seen here. Then, if you see the characters here, the characters are controlled by discrete unit called factor. Mendel gave the term factor. So, we are going to use the term factor here. It's nothing but gene. Then factors occurs in pair. So, how do they occur? Like TT, which represent tall, and small t, which represent dwarf. In a dissimilar pair of factors, one member of the pair dominant while other is recessive. Here we are going to say the term heterozygous, capital T and small t. I'll write once again, capital T and small t. So this is dissimilar pair. One is dominant, another one recessive. But when you say this heterozygous by phenotypic expression, you say again it has a tall. It is not a dwarf because the T, capital T, which is the dominant character, will mask the small t, which is recessive character. Law of segregation, the second law. the two copies of each chromosome will be separated from each other, causing the two distinct allele. So if this is the chromosome, okay, pulling apart, okay, like this, pulling apart the chromosome, and if you are going to write the gametes like, the alleles like this, you can pull out like this, capital T, okay, segregation of the gametes from each chromosome. So now we will see the monohybrid cross in detail. I am going to use the size of the plant here. Mono, when you say monohybrid, one character, okay, one character I am going to take. Hybrid, I am going to get a combination in that. So first I am going to write the parents tall plant, another one dwarf plant, okay. Tall plant I am going to represent with the letter capital T, which you call it as genotype, and then small t, which you call it as again genotype for the dwarf. Okay, this is phenotype and this is genotype. You have to write both the things. Then when it is intercrossed, okay, before that you have to pull out the gametes like this, capital T, and encircle the gametes small t, okay. And this you call it as G, nothing but gametes. Then next, this capital T and then this capital T, small t, will together form like this heterozygous condition, and this again represent tall phenotype. Again if you intercross this, you will get like this. So here totally 100% you will get tall. But it is a homozygous or heterozygous? It is a heterozygous condition. So this you call it as F1 generation or the first filial generation. There was another scientist called Punnett. He introduced a simple method to represent this and to calculate this. That you call it as Punnett square or checkerboard. That I am going to show in the F2 generation. So I will repeat again, parent and the two parents we are going to intercross. And before that we will pull out the gametes, and from here if you intercross you will get a character. What character you are getting? The dominant character. So it obeys the law of dominance. So first law, it is obeying. Then next you see segregation of the gametes. So it also obeys the second law of segregation. In board based question, suppose if they are asking you to explain the law of segregation by giving an example, you can talk about the monohybrid cross. Dominance, both you can talk, monohybrid as well as dihybrid. Okay, we will move on. F2 generation, here one more thing, on self. You have to write on self. We are going to intercross this on self. Okay, both we are going to intercross, and that will become the F2 generation, these offsprings. So here is the Punnett square or the checkerboard, and here you can write the symbol for male and female, okay, and here it won't show any difference in this. When it comes to any X-linked inheritance, there shows much variation. So here you can write anywhere male and female, because we are going to use the same genotype capital T, small t, okay. So we will use capital T, small t, capital T, small t. So what is this capital T, small t? The same thing, okay. This part, I am taking this part and I am writing there, okay. We are intercrossing the F1, self, okay. So now this capital T and this capital T together I will write, they will become the allele, they will become the genotype, and they express your phenotype. So what is the phenotype here? It is tall. So we will write that also, tall. Okay, then next, first let us complete the pairing, capital T, small t, and this capital T and small t. Again this small t and small t. We will write their phenotype. Now genotype is ready. We are going to check their phenotypes. So what will be the phenotype? Capital T, small t, homozygous or heterozygous? This is heterozygous. So it is tall. And this is also tall, because the dominant capital T will suppress the character, recessive character dwarf. And this is dwarf. Okay, now it is ready. We are going to check about their phenotype and genotype. In the previous first generation, we got 100% tall. But the condition genotype, if you take capital T, small t. Let us see here with the ratio. So first, we will talk about the phenotype. Okay, the phenotype. So we have tall as well as dwarf. In what ratio we are having? Tall, how many talls are there? 1, 2, 3. So we will write 3. How many dwarf? 1. So 3:1. Out of 4, you are getting 3. Okay, we can write like this also. Out of 4, we are getting 3 tall. And then out of 4, we are getting 1 dwarf. Okay, in a fraction. And in percentage, if you take 75% tall and then 25% dwarf. Okay. Now the genotype, we will see the genotype. Genotype. So what are the 3 genotypes we are getting? Capital T, capital T. You can see here, and then capital T, small t, and then small t, small t. Okay, 3 different genotypes we are getting. Okay, in what ratio we are going to get? That let us check. How many capital T, capital T? 1. And how many capital T, small t? 2. Okay, and let me encircle so that you can identify 2. And how many small t, 1. So 1:2:1. This is very, very important 1 mark question or 2 mark question also, totally the cross, and the ratio is very important. So concentrate in the ratio. Now let us see the percentage. This is 25%, this is 50%, and this is 25%. Okay, and if you want the fraction 1/4, 2/4, even that let me write 1/4, that is out of 4 1, and out of 4 2, that you can write like this 1/2 is 2, and 1/4, out of 4 1. Okay, so now we know the phenotype and genotype. Let us move to the next cross. What is the next cross? Dihybrid cross. So once you know this clearly, dihybrid will be very, very easy for you. So next, it obeys the law of independent assortment. So what is independent? The traits inherited through one gene will be inherited independently. Okay, trait, we are talking about the character. Suppose the color of the plant, if you are going to take it, and color, another one also you will take size. 2 traits I am taking. One is the color, another one the size of the plant, let me take. Okay, both the character will show their presence independently, will not depend on the other. Whereas when you take like this, one single character when you take, they are dependent. Okay, but here when you take 2 different characters, they are independent. Okay, through another gene, because the genes reside on different chromosome. This is the reason why it is happening, why they want to show independently, because they show, that is, they are there in the different chromosome. Okay, that is why you call it as independent assortment. And here I am going to talk about dihybrid cross. Okay, so in dihybrid I am going to take 2 parents, 2 characters, 2 characters. Okay, hybrid, you are going to get the combination. Okay, so here parent, if you take 1, I am going to take size. Okay, or else we will go according to our NCERT. In NCERT, if you see, that is color, yellow color. Okay, shape, round, like this, yellow color. Another one, shape. Okay, so shape is round, color is yellow. Okay, round, yellow. Another one I am going to take wrinkled green. Okay, this I am going to indicate by small y, small r. Okay, the same thing I am going to take there. So listen, so here parent, capital Y, capital Y, capital R, capital R. Small y, small y, small r, small r. Let me write the phenotype also. What is the phenotype? This is yellow, round, and this is green, wrinkled. Okay. Now we are going to pull out the gametes. This is very important part. So listen how to pull out the gametes. We are using two different characters. So here capital Y, one character, another character capital R. So two characters individually you want to take, because those two characters only will be inherited to the next generation via chromosome, right? Next, capital Y, capital R. Encircle the gametes. Small y, small r. Small y, small r. And it is not a biology thing, okay, a small trick I will tell you. When it is coming in 4, okay, a pair with another pair. Okay, when it is coming in 2 different letters. Okay, pair with pair 4, 2 different letters in a pair. Okay, like that just you keep in mind. And now this is a gamete. So we have written the gamete part. And here we are going to see the cross now. Okay, let me intercross now, and this is F1 generation. When you intercross, you may do mistake by writing YR, YR. But you have to write like this, capital Y, small y, capital R, small r. Okay, 2 letters. Then only it will become allele and it will express their character. Again, 2 letters in a pair. It's same letter only. Then only it will express their character. Now you have 2 characters. What are the 2 characters? One is the color, another one the shape. So, okay, YY, RR. The same thing here. Capital Y, small y, capital R, small r. Okay. So, we get something dominant we are getting. Just see what is dominant character expressed here. So, it is yellow, round. We get yellow, round. 100% again. In next generation, okay, when I self this one, on self, okay, on self, what will be the ratio? What are the different characters? Okay, to the surprise, we are going to get 2 different characters apart from the parental character. So, what is the parental character here? Yellow, round, green, wrinkled. But we are going to get, okay, 2 different characters, yellow, wrinkled, green, round. Okay, in what ratio? So, let us see that one. Okay, the same thing. I want to pull out the gametes. Okay, we have capital Y, small y, capital R, small r. Okay, let me pull out the gametes. When you pull out the gametes, capital Y, capital R, capital Y, small r, small y, capital R, small y, small r. These are all the 4 gametes. In dihybrid cross, you will get 4 gametes. In monohybrid cross, you will get 2 gametes. So, keep that in mind. Again, represent with the symbol male and female. So, we will write the character capital Y, capital R. Okay, the same thing. You can see here these 4 gametes I am writing here, male and female. Then capital Y, small r, small y, capital R, small y, small r. Okay, the same thing. Another one also, if you see here, this is also the same thing, and this one also the same hybrid. Homozygous or heterozygous? It is heterozygous. Okay. Capital Y, small r. And then you can write small y, capital R, small y, small r. Okay. And now I am going to write it in pair. Capital Y, capital Y, capital R, capital R, capital Y, capital Y, capital R, small r. Okay. I am writing these 2 together. When you are writing it together, it should be in a pair. Okay. So, keep that in mind. Capital Y, small r, capital R, capital R. Same thing. Capital Y, small y, capital R, small r. Then capital Y, capital Y, capital R, small r. Capital Y, small r, small r. Then capital Y, small y, capital R, small r. Capital Y, small r, small r. Same you complete with this. Capital Y, small y, capital R, capital R, capital Y, capital R, small r, small y, small r, small r, small y, capital R, small r. Okay, capital Y, capital R, small r. You have to check any mistake is there. If there is a mistake, you won't get the ratio. Then capital Y, small r, small r, small y, capital R, small r, small y, small r. So we got perfectly the genotypes. Now we are going to represent their phenotypes. So this is, let me write YR alone, yellow round, okay, yellow round, because we have dominant character, yellow round. Again here yellow round, yellow, two recessive character, so it is wrinkled, yellow round, yellow, these two you call it as wrinkled here, okay, wrinkled, yellow round, green, this is green and this is round, okay, and here again it is green, it is round, capital R is there, so it is round. Then yellow round, yellow wrinkled, we have two recessive character, here green round, here both are recessive, so green wrinkled. Okay. Now let us see the phenotype and genotypic ratio. Phenotype. Okay, phenotype, what are the phenotypes we are getting? We will check. One is yellow round, is 2, and then new character, any new character, yellow wrinkled, anywhere you are getting? Yes, we are getting yellow wrinkled. Okay, let me show this yellow wrinkled. Okay, then green round, again new character we are getting, green round, and then green wrinkled. Okay. So let me see the number. What are the numbers? Yellow round 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 is 2. Yellow wrinkled if you take it 1, 2, 3. 3. Green round you take 1, 2, 3. Green wrinkled 1. Okay, this is the phenotypic ratio. This is another important topic. Okay. So next class, okay, I'll pose a question here. Try to find out the answer. We'll see in next class. Okay. To receive our online lessons, please press subscribe button and you will receive the latest updates. Thank you.